Okay, welcome. So we are going to do a multiple choice. It is October, November 25, so a fairly recent one. As I'm sure you already know, it's a 40 question paper. You get 45 minutes, so slightly over a minute a mark, very generous. The way I would always encourage you to do a multiple choice is to try and eliminate. Sometimes you'll just know the answer straight away because they're more straightforward. Some are not. They like their table format. Cambridge do. So sometimes it's a case of eliminating across the rows, as I'll show you on some of the questions as we go through. As I'm going to do a bit of teaching as I go through the questions, just to kind of help with your general knowledge and understanding across the whole of the course. I will just do 10 questions at a time rather than do all 40 in one go, because otherwise it might be a very long video. What was I going to say? You would get, when you sit this, and I'm sure you've probably already done this as a mock at this point, earlier in the year, you would get an answer sheet where you mark in pencil your correct choice, so between A, B, C and D. You don't actually do it on the question paper. Obviously, if you want to write on the question paper to help you, I'd really encourage that. Okay, there's nothing wrong with doing that. Obviously, you just don't write your answer on the question paper because it needs to go on the mark sheet that then Cambridge obviously gets sent to the exam board to mark. Okay, so we're going to do the first 10 questions. The great thing about multiple choice is it does go in order of the topics of the syllabus, so it can really help you audit your knowledge and understanding if you're doing these for exam prep, and also for the theory papers as well, just knowing where your kind of weaker areas are, so the topics you want to go over more and look at again. And then also it might give you a bit more confidence in those topic areas you feel like you've got it, you know the content a bit better. So we normally start with Mrs. Gren question, so this is quite unusual. Mrs. Gren obviously being the kind of 7 characteristics that all living things have or show. This is actually starting with a bit of classification and how you name organisms. So it's about the binomial name, bi meaning two, nom or nomial meaning to do with the name. So we never just give an organism its species name by itself. We always pair it with its genus name. So the Rattus norvegicus, or norvegicus, and the Rattus rattus are species names, but incorporated into it. The first part of the name with a capital letter is its genus, and then the second part is its species name, which is given a lowercase letter at the start, and it's written in italics when it's typed, underlined if it's handwritten. Okay, so that's just universal. Everyone around the world, it doesn't matter what language you speak, it's normally based in Greek or Latin, which are archaic languages. They've been around a very, very long time before modern language developed. So, which statement about the rats is correct? The rats belong to the same genus is our first option. I know straight away that is the correct answer because the rattus part, the genus part of the name, is the same. If I just check through the rest of the options just to be sure, they belong to the same species. No, they've got a different species name. The second part of that binomial name is different, so that's definitely a no-no. C, they can breed together to produce fertile offspring. That's more asking you about what we mean by a species. If they're not the same species, which they're not because they've got different names, they might be able to breed together potentially, but they won't produce fertile offspring. They'll be sterile. So in this case, it's a no to that. They can't breed together and produce fertile offspring, which is what makes an organism belong to the same species. And the fact that D is the rats do not share any of the same features. Well, we can look at the two images and go, well, they're very similar. We could hardly tell them apart, really. So that's not true either. They do share many similar features. They're just, they may be different, obviously, genetically by a small degree, which we can't necessarily see with our eye. Okay, so moving on, that was 1 is A for alpha. So 2, we're looking at kingdoms now. This is quite a tricky question, really. Cambridge don't really go in easy. They kind of start difficult right from the beginning of a paper, and they don't really ramp it up at all like some exam boards might do. So we've got different organisms. My eyes are drawn to cell 2 right in the middle there. I'm looking at that straight away and thinking, well, that's a bacterial cell. I can tell it's got this whip-like extension called the flagellum. It's also not got a nucleus, whereas the 2 cells either side, you can sort of see that perfect black circle, which represents the nucleus where the DNA is found. You'll see in the bacterial cell in the middle, there's no nucleus. Those small black blobs, by the way, I think are supposed to represent ribosomes, although they are very, very small in reality. And then the big squiggly kind of line in the middle, well, it's a big continuous loop really, but it's just all tangled up. That's its circular chromosome, so that's its DNA. But yeah, it doesn't have a nucleus, unlike cell 1 and cell 3. So if I look at the table without actually having to do a lot of work, cell 2, if I know that's a bacterial cell, I should know that belongs to the prokaryote kingdom, meaning before nucleus, so they don't have a nucleus. So actually the only option that gives prokaryote for cell 2 is line B, so or answer B. So actually I'm happy with that. I'm just going to check either side. So cell 1 is a fungus. If I look at cell 1, it's a single-celled organism. It's got a cell wall, which I know fungi have. It's got mitochondria, which there's those sort of 6 larger looking structures with the inner membrane folding inwards. That's the mitochondria. That's where respiration takes place. It's got a nucleus. I'm happy with that cell 1 being a fungus. And then cell 3 looks like an amoeba to me, which is belonging to the Protoctist kingdom. Again, it's a eukaryote in that it's got a true nucleus, and it's got cell structures like mitochondria, which the bacteria is quite unusual. Being a prokaryote, it's not evolved to have those same structures. It's a much more simplistic organism. Okay, right, and a lot smaller. Although, as you can see, it's not to scale, so that's not obvious on the actual drawings. Right, so that is question 2. We are going for B for Bravo. Let's move on. So question 3, we're still moving away from classification more to cell biology now. So structures of a cell. I have been given a plant cell diagram. The biologist wants to find out the number of chromosomes it contains, which labelled parts should be examined more closely. Well, chromosomes are how your DNA is organised. We know the DNA is found in the nucleus. The nucleus would be part A for alpha. Just going through the other cell parts is a bit of teaching. B is the central vacuole there, so that's where it stores the cell sap, so water with salts and sugars and various other things dissolved that the cell may need. It's kind of storage. It also helps push out on the cytoplasm, which then pushes out on the cell wall and makes the cell turgid for support of other plant tissues. C would be cytoplasm. D would be the cell wall, which obviously gives that kind of rigidity and the shape to the cell, and kind of stops it from bursting as well. So we're going for A for alpha. Right, question 4, we're looking at organisation of the organism. So it lives within that sort of cell structure topic, it builds on it. So cells are kind of the simplest structure, and then they are collected. They are not collected. They are organised into tissues. Then tissues organised together, performing a function, are organs. Then the organs grouped together, performing a particular function, is an organ system, and then all the organ systems collectively is the organism. Okay, so that's how the hierarchy works. This is fairly straightforward. I mean, I would hope most people know the brain is an organ. It's definitely not a tissue level. It's a very complex structure. So I know it's going to be A and B. I can eliminate C and D, hopefully, not have to look at those again. So I'm just looking at A and B now in the table. The liver, also a complex organ. It does a lot of things in the body. I'd be thinking straight away an organ. So for me, A would be my choice. And then the neuron is basically what we call a nerve cell. It's the correct term for a nerve cell. So yeah. So basically the difference is the middle column anyway, knowing that the liver is at an organ level rather than a tissue level. So A for alpha. Right, 5, we're moving on to a bit of biochemistry in the form of DNA. So DNA is our genetic material. It carries the code for proteins that our cells need to make. It's a double-stranded molecule. We have been given one strand, and it's asking us what would be the complementary strand that pairs with that. So the way I teach this and the way I remember it is apples and trees. So A and T go together, cars and garages, C and G go together. So this is quite straightforward. If we read across the strand, it would go complementary. We would have a T going with the A and G going with the C. Now, if we look at our options, there's only actually one strand given that has T and G at the start, and that's letter D. So we know it's D for delta. Okay, A is just option A is just repeating the same strand. Obviously, that's not complementary. So D for delta for question 5. Right, question 6. Bit of biochem still. Which substance is an enzyme? So, not a trick question as such, but most enzymes end in A's, so ASE. However, not all do, and pepsin is the one that doesn't. It works in our stomach, it breaks down protein. It's an unusual enzyme because it works at low pH, whereas most enzymes kind of work around neutral or slightly above. So it's C for Charlie. Just running through the other substances so you know what they do. Fibrinogen, you might recognise it, hopefully you do. It's involved with blood clotting, so it helps the blood clot when we kind of injure a blood vessel, stops us obviously bleeding out, stops naughty pathogens from getting in. Messenger RNA is a molecule that it's basically, rather than the DNA moving out of the nucleus to a ribosome for when proteins are being synthesized, the messenger RNA is the molecule that takes the code, takes that genetic information from the DNA kept safe in the nucleus to the ribosome, and then the ribosome can then make the protein using that code. Obviously pepsin is our option there, so we know that's the enzyme. And then D, saliva. Saliva is not an enzyme in itself, okay? It can obviously have an enzyme called amylase within it when it's secreted into the mouth for digestion of starch, but saliva itself is not the enzyme. All right, so happy with that. 6 is C for Charlie. Okay, 7, we're moving into human nutrition. Which function is performed in the duodenum? So, knowing your alimentary canal, the duodenum is the start of the small intestine, so it follows on from the stomach. And what's really primarily is happening in the duodenum is basically loads of digestion, because many of the enzymes that are involved with breaking down the various components of our diet are working in the duodenum. So we've got many of them are being made by the pancreas, which then is released into the duodenum down the pancreatic duct. Some of the enzymes themselves are actually made by cells that line the duodenum. Okay, so we're going for digestion. All right, so that's B for Bravo. Just to go through the other 3 terms though, as I said, I'll do a bit of teaching as we go through. Assimilation is basically what happens with what you've digested, absorbed into your blood, and then what the cells do with that. So they might store it away. So for example, they might store glucose as glycogen because it's not all needed, and we don't want too much glucose to be travelling around in the bloodstream because it can cause problems. We might make it into something different. So the amino acids that are absorbed and taken up by cells might be built into brand new proteins, because a lot of what we basically absorb is the building blocks for new molecules within our cells for growth, repair, etc. Egestion is to pass out feces, so that happens at the anus. That's to get rid of all the kind of non-digestible matter, the fibre, various other materials that pass through and don't actually get absorbed. And then de-ingestion is what we call the process of actually putting things into the mouth. So when we effectively eat and drink, that is ingestion. Okay, let's move on. So just to say again, 7 is B for Bravo. 8, this one is probably looks a bit to some people because it involves a graph. Not everyone loves a graph. If we just look at it, it's quite straightforward. It's just showing you as food travels through 3 regions of the alimentary canal. So the esophagus is what connects the mouth to the stomach. Okay, so basically some people might call that the food gullet. Then you've got the stomach, and then you've got the duodenum, which we were just talking about previously, which is the start of the small intestine. We can see the pH is not quite neutral in the esophagus, slightly, I wouldn't say acidic, but just under a neutral pH. Then it drops to quite a low pH in the stomach, and then it goes back up again. It goes slightly overshoots neutral to slightly alkaline in the duodenum. So we're looking for the substance that causes the change in pH at the point labelled X. So we look at X, we're seeing an increase in pH. So we're going from acidic to just over neutral, slightly alkaline. So we want an alkaline substance to do that. Obviously, it's going in the opposite direction to what happened from esophagus to stomach. So the alkaline substance in the list is bile. Bile is made by the liver, stored in the gallbladder, and then secreted down into the duodenum via the bile duct. Okay, so yeah, it is alkaline. It also is involved with the emulsification of fats, which is another role that it does. Gastric juice, so B, gastric juice would be very acidic, so it wouldn't bring the pH up, it would keep it low, so it's not that. Glucagon is a hormone that brings your blood sugar level back up again when it gets too low, so that's got nothing to do with this. And trypsin is an enzyme that digests protein in the duodenum, so it doesn't change the pH as such. So we're looking for bile, so 8 is A for alpha. Okay, 9, we're still on human nutrition. They've gone big on the human nutrition topic in this particular paper. Which row names a small molecule and where that molecule is absorbed in the digestive system? All right, so much of the food stuff that's broken down is absorbed in the small intestine. So if we look at amino acid, which is the breakdown product of protein, it's saying no in the small intestine, but yes in the colon. Well, that wouldn't really make sense, because actually it would be the small intestine where it's absorbed, and actually the colon is the large intestine, where that's not really about absorbing the breakdown of food in a great deal, to be honest. So no, A is not right. B, we've got the same problem. Glucose is mainly absorbed in the small intestine, and again we've got a cross for that, so that can't be right. So let's get rid of A and B to those two. Maltose, small intestine, yeah, okay. So maltose, okay. Maltose, the reason why that is wrong, little trick there. Maltose is not absorbed as maltose, it's still too big. Maltose is actually two glucose molecules bonded together, so it needs to be broken down. So C is a red herring, all right, in the terms that it's not a small molecule. It's what we call a disaccharide, so it's a sugar that still needs to be broken down to its simplest form, which is glucose. So no, C is out. And actually the answer, this is one where I think many people would probably get it wrong, it's D, it's water. Majority of water is actually absorbed in the small intestine, and then there is more reabsorption of water in the colon, so we don't actually pass out too much water in our faeces, because then unfortunately we would have diarrhea, which is not ideal and can be fatal if it's obviously not kind of treated. Okay, so 9 is D for delta. Right, 10. Okay, so we're moving on to what looks like a bit of plant transport, so how plants move materials around their body, or their structure, if you like. So this one is where we probably need a bit more time to look at what's been given to us, because we've got a little experiment, a little diagram to analyse. So it says a student cut one stalk of celery from a celery plant. They put the celery stalk into a beaker of blue dye. The celery stalk was removed from the blue dye after 2 hours and was cut into sections. You may have even done this fun practical yourself once upon a time, but what is shown by the blue circles in the cut sections of the celery stalk? Now, it's water that's moving up the celery stalk, and it's been dyed blue. Okay, this is quite hard. People find plant transport tricky. What you should be thinking is which bit of the transport system carries water, and it's the xylem. All right, so the xylem carry water and mineral ions, but obviously it's not really asking about mineral ions, it's just asking you about water that's had a bit of blue dye dropped into it. So if we look at our options, it would be D for delta. All right, not phloem. Phloem carries sucrose and amino acids, so not that. Cortex cells are the cells that kind of form the rest of the kind of packing material that's in the stem. All right, so not the actual transport vessels or the kind of vascular system. It'd be the rest of the material that you would find in that stem. Palisade cells, the palisade cells are found in the leaf, all right, so they're nothing to do with the stem, so don't worry about that. They're just throwing you some odd, it's hard, people find plants harder than animals. All right, now I'm going to stop at this point because that's 10 questions done, and then I'll do the rest of the paper in three separate installments. All right, thank you.